Hello, everyone, and welcome to the first video on translation. In this video, we're going to do just an overview of the process, and then there'll be a second video that's looking at specifically what is happening every step of the way. So, this process is called translation, and the name is important, y'all. This is probably a word that you have heard before. And so think about when a translation is needed, right? When you have one language, but you need to be understanding a different language. And that's going to be what's happening here. Because with translation, what's going on is the cell is going to be using RNA to make proteins. And so we also refer to translation as protein synthesis because that's our end goal. We are making or synthesizing proteins. So remember, this fits into the overall process of gene expression. So remember, gene expression is taking the instructions found in DNA and using them to make proteins. So in the last video, we talked about transcription, which took DNA and made RNA. Well, now we need to translate that RNA so that the cell can make proteins. Because RNA is made up of nucleotides, but proteins are made up of amino acids. Very different things. Okay, RNA has 4 different nucleotides, A, U, G, and C, but there are well over 4 different amino acids. There's over 20 amino acids. So there's going to have to be some sort of translation device to help us go from an RNA sequence to a protein sequence. And so what's going to be happening here is those RNAs that we talked about in transcription, rRNA, tRNA, and mRNA, are now going to be put to use to help build the protein. So, translation is going to be happening in the cytoplasm of our prokaryotic cells. Because remember, they don't have any internal organization. So, this will be happening in the cytoplasm. If we want to be more specific, it's happening in the cytosol, that jelly like fluid that's occupying the cytoplasm. And here we have 3 key players, but there's kind of like a hidden 4th key player in there. So remember, this is where all that RNA that we made during transcription is going to come into play. So our first key player is the mRNA. So remember, M stands for messenger. And this is important, y'all, because the mRNA molecule serves as the message, the directions that the cell is going to use directly in order to build their proteins. Okay. And then the mRNA is going to say, hey, here's the directions. You need this amino acid first, you need that amino acid second, and then we'll need, blah, 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 third, right? And it's going to go on for about 500 or more amino acids. So the mRNA is just saying which amino acid is needed. The tRNA, and the T stands for transfer, because the tRNA is the gopher that's going to say, oh, okay, we need this amino acid first. Let me go find it in the cell and bring it to this place so we can build the protein. Okay, so the tRNA is the gopher that's going out and finding the amino acid that's needed and bringing it to the site where proteins are built. Well, where is that site? That's going to be the ribosome. Remember, our ribosomes are protein making machines, and they serve as the factory, the physical location where the amino acids are going to be brought and connected together or bonded together. Okay, so who's our kind of hidden 4th key player here? That's going to be the rRNA. Okay, and the R in rRNA, let me write this down, equals ribosomal. Okay, and so even though in this picture of the ribosome, you think it's made up of only two pieces, a small subunit and a large subunit that come together to form a functional ribosome. And while those steps do happen, okay, there's also ribosomal RNA, and it's actually the core structural and catalytic component of the ribosome. Okay, so it's not shown in a lot of the pictures, and I think it's just for simplicity, but the rRNA really forms like this physical backbone of the small and large ribosomal subunits. It's going to help create the specific binding sites, and you can kind of see these sites right over here. Okay, and this is where amino acids and things are going to go, and they're going to be linked together. So the ribosomal RNA does have a really large part in that. Okay, but because it's three separate pieces, the rRNA, the small subunit, and the large subunit, in pictures, it tends to just look like the two subunits. Okay, but the ribosomal RNA is in there. So what we made in transcription is coming back to help us in translation. Now, the other thing that I want to point out to you is this need of a translation device. How are we going from messenger RNA, again made up of nucleotides, to a protein made up of over 20 different amino acids? Well, what happens is the messenger RNA is going to be read in sets of three nucleotides, and we call that a codon. And so you see that right here. And so when you have an RNA sequence, you just divide it into sets of three. And again, each one is called a codon. And then I'm going to provide you with a codon wheel, which you'll see here in the next slide, that translates it. It said, okay, if you have these three RNA nucleotides, you need this amino acid to build your protein. It's really pretty snazzy, y'all. So here's how I remember this. The mRNA is the messenger RNA. It's made up of codons. So I like to say that mRNA is actually a coded message. And that helps me remember that it's the messenger RNA made up of codons. Okay. And that distinction, remembering that codons are on mRNA, are going to come into play a little bit later. But first, I want to kind of show you what's happening during translation so you can see really the different roles of these key players. So what's going to happen, I'm actually going to go into animation mode, is what you see on the right here is the codon wheel. That's the translation device that's going to help us go from mRNA to amino acids. So we have our mRNA sequence here, and had I not labeled it, you would have known that it's RNA because it runs 5' to 3', and it has uracil. It has the base U. Remember, you do not find uracil in DNA. So if I just showed you a picture of this sequence, you would say, aha, there's a uracil, so I know I'm looking at RNA, and then the 5' to 3' would confirm that because that's how RNA is made. Okay? All right, so if I gave you this sequence and I said, I need you to tell me what the amino acid sequence is going to be based off of this message, what would you do? Well, the first thing you want to do is always divide your messenger RNA into sets of three, into codons. And I space it out for you this way for this particular slide, but normally the letters are all right next to each other. Okay, so you are going to identify the codons. The first codon is always called the start codon, and it is going to be AUG. That's just how it is. That is our first codon, which is known as the start codon. So then notice I have all the other sets of three, the other codons identified, and we just number them. So we have codon 2, 3, 4, and 5. And then the last codon also has a special name called the stop codon. And so all we have to do is we use our codon wheel and start with the first letter of the nucleotide. So I'm going to start right here with A, then I'm going to move to the U, and then to the G. So when you look at this wheel, you always start in the center. So my first nucleotide is A, so I'm going to start right here. And then I'm going to move. So now you see the other 4 bases right here. So now I'm going to say, okay, my second one is U, so I'm going to go this way. And then there they are again. And my third nucleotide was G, so that brings me right here. Methionine is my first amino acid. Okay, now you don't need to know that MET stands for methionine on the exam or knowledge check or whatever. You would just put MET, just the 3-letter code. All right, so now we're going to do the second one. So CCA, so again, you start with your first nucleotide here, C, work your way out, CA is going to take us here, whoops, to proline. Okay, now let's do the next one. We're going to go GUA, so we're going to, start here at G, work our way out to U and to A, and we get valine. All right, so I'm going to erase my doodles, but go ahead and try to finish at least the next 2 and tell me what amino acid you're going to get from those particular codons. All right, so GAA, we're going to start here at G, work our way out to A, and A, we're going to get glutamine, GLU. Now we're going to move on to UCC. UCC gives us serine. And then the last one, our stop codon, the name is kind of a hint. It says, hey, we are done. And if we go to UGA, look, we have this circle. So go down to our key, and that says stop, which is probably not a surprise because it was called the stop codon. Now, notice that the word stop is not part of this wheel, right? There's no 3-letter code. So stop is not the name of an amino acid. And y'all, with names like methionine, proline, valine, glutamine, do you think we're going to have an amino acid called stop? No, we don't. So why that's important is you don't write anything. Okay, there's a whole process that happens when this stop codon is reached that tells the cell, we're done, protein's complete, the end. So if I ask you for an amino acid sequence, which I will, do not write the word stop there. You're just literally done writing. Okay, y'all? And so this is animated so that it would have been a little cleaner. So let me go ahead and erase what I did because I want to go back and just walk through this process one more time really quickly. So again, we're giving you the mRNA sequence. You know it's RNA because it runs 5' to 3' and because there's uracil. To figure out your amino acid sequence, you're going to divide your RNA into sets of 3 called codons. The first one is called the start codon. Then we just number them, and then the last one is called the stop codon. Okay, so then you're just going to go ahead and use this codon wheel starting from the center and working your way out to figure out what those amino acids are. Okay, so remember if we start and we go AUG, we get methionine, CCA is proline. Okay, and then you're just going to keep working your way out. And then when you get to the end, there's no amino acid called stop. Oops, sorry about that, y'all. And we are done. Okay, so that's going to be the end of this video. But in the next video, we're going to talk about how we actually get a methionine that's in the cell and bring it to the ribosome, the factory. How do we then get proline next? And how do we make sure that proline and only proline is delivered second, right? Because the cell has hundreds and hundreds of different proteins. A lot of them are being made around the same time. So how can we ensure that the correct amino acid is brought to the correct spot at the correct time, right? There's a lot of things that have to happen for that. So get excited. We're going to talk about that in the next video. Bye, y'all.